Excellent. Well, thank you, everybody. So I'm presenting actually on behalf of Varuni, who's a trainee in Sergey Mangul's lab, who wasn't able to come because she got an internship. So I'll be presenting on her behalf about TAMPA, a way to do profiling visualizations. So the context here for this tool is taxonomic profiling. So taxonomic profiling, we've seen that a few times today, but basically you're taking your raw metagenomic reads and then paired with some sort of reference database. So in this case, a bunch of reference genomes. You want to be able to get a list of the different taxa that are present in your sample and the relative abundances. So with this, you can generate things like this little pie chart telling you which taxa or who's there and how much. So the motivation, or I guess the thing that I want to focus on here is this little format for the taxonomic profile, which actually has a story behind it. So before CAMI, which you may have heard of CAMI, which is Critical Assessment of Metagenome Interpretation, there was a plethora of different taxonomic profiling tools, things like Metafiler, Metaflan, yada, 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 bunches of different tools. Each one of these had all different output formats. That can be a major headache when trying to do tool development and tool comparison and benchmarking, like what CAMI is trying to do. So what the CAMI competition did was require a uniform standardized output format so that we can easily compare these different tools. So post CAMI, we now have this standardized output. You know what they say is a great thing about standards? There's so many to choose from. No. So in this case, the nice thing about a common standard, is that an ecosystem of tools can be built and that can be applicable to a bunch of different taxonomic profilers. So as an example of the kind of ecosystem of tool that is going around this effort of trying to at least standardize output formats are things like CAMI Sim to simulate metagenomic communities, CAMI Tax to harmonize taxonomic labels, so we're all talking about the same taxa at the same time, and then benchmarking platforms like Opal, which allows you to generate figures that look like this. So in this diagram here, we have a gold standard, which corresponds to a simulated data set, and then we have a whole bunch of different tools up here. And then we have some common metrics that are agreed upon by the community to measure the accuracy of these taxonomic profiles. That's great. These numbers are nice. However, when you end up with something like this down here with a weighted unifrac, and you see a value of 7.65 and 7.79 for two different tools, what does that mean? So these are great for ranking, but what is the actual biological differences, if any, for the difference performance of these tools? So what we would like to do is a way to actually compare and visualize the outputs of these tools instead of just looking at their metric values to try to get some more biological insight. So that's where TAMPA comes in. It's a nice little tool to visualize the outputs in the standard CAMI format. It's inspired by Metacoder. I don't know if you've heard of Metacoder before. It's been used in Amplicon and 16S RNA studies. They make these nice plots like that. So the thing is that it's very much tied to an Amplicon kind of approach. That's why you see an OTU count right here, and we're doing whole genome shotgun metagenomics. But so we wanted to take this general idea and then apply it to whole genome shotgun. So inspired by Metacoder, we can display the abundances and quantitative information on the actual taxonomic tree. So this is an output of TAMPA, and you can see here that we are comparing the outputs of Metafiler compared with the ground truth. And the little discs here on the tree represent the relative abundance in one of the samples. And then you can see that each one of these little discs is kind of like a pie chart. And that's going to tell you the differences in relative abundance between the couple of tools that you're comparing. And then also some of the text here is in blue bold, and that is emphasizing significant differences or differences above a user-specified threshold. So what are a few different use cases for TAMPA? So one is that you can compare the output of your tool, like if you're a tool developer, or if you want to see how good a tool is on some simulated data set, to the ground truth. So an example would be like this. We have Metafiler again being compared to the ground truth. And this can really help us then dig down to what the actual biological differences are in the benchmarking platforms like Opal. So here's that little one specific column of Opal here, and you can take a look and you can see here, oh, we have an L1 norm of 1.13. What in the world does that mean? How good is that? How bad is that? Now we can actually see a little bit more emphasis that, oh, metafiler has predicted this chloroflexy taxa, which is not present in our ground truth, and that's contributing to the L1 norm error. So it really helps elucidate some of the biology behind the metrics, which can be handy for cases like this. So here are two taxonomic profiling tools, TaxiPro and Metafiler. And two of the metrics, or one of the metrics that Cami looked at is this unifrac error. And it actually turned out that these two tools had the exact same unifrac error. So you'd think, oh, they're performing the same. Well, now that we actually visualize it and we take a look and we can see, oh, they actually have very different taxa that are being predicted here. And so it can help you understand the biology that's happening behind the metrics. It also helps visualize global tool performance. So this is a nice little analysis. So we use this tool called MIT that identifies the most difficult to identify taxa. So we took all of the Cami simulated data sets. We looked at all, two dozen different taxonomic profiling tools, and ran some stats on that to figure out which ones of the taxas are most difficult to identify. And so we get this nice little list. And it's like, well, I want to visualize that list now. So you chuck into TAMPA here, and then you can see that, oh, okay, so these here are the most difficult to identify taxa. And so you can get some interesting ideas. So, oh, hey, look, plasmids and viruses are some of the most difficult to identify taxa amongst all of the taxonomic profiling tools that were submitted to the second Cami challenge. And so if you're a tool developer, yeah, maybe make sure those are included in your reference databases, including like right here, we can see that all these guys are archaea. They are also difficult to classify. But beyond doing benchmarking and simulated data sets, we can also compare outputs on real data. So I just went and pulled some random study looking at patients with Crohn's and patients without. And we can do a linear plot of the taxonomic profile. I did it with MOTUs or MOTUs. And we can see a lot, if we plot it in a linear relative abundances, a lot of Bacteroides. But if we take a control patient, we can see here a lot of Firmicutes instead. And then if we want to compare the two against each other, and then like do it with a log plot to be able to see the differences, you can start to really visually see the story that the authors in this particular study were trying to show in the differences in which bacterial organisms are most prevalent in gut microbiomes of patients with and without Crohn's. So you may be asking, well, how is this different from some of the other visualization tools that are out there? So you may have heard of Krona. So Krona is this nice way that you can get these sort of hierarchical pie charts of any sort of hierarchical information. It doesn't have to be a taxonomic profile. But well, this is great. You would have to do a format conversion because it doesn't take Cami input. And the one difference between it and TAMPA is that Krona does a single sample, whereas we wanted to compare two samples against each other. And then there's Graphland. Graphland makes these beautiful plots like you see here, and you've probably seen in some publications. One unfortunate thing about Graphland is it's tightly integrated to a single profiler. And it's very difficult that if you take a different tool to try to get it to work in something that Graphland would like. So that's why we targeted the Cami format, because, well, 30 some odd tools have already standardized on that output. So might be more probably useful that way. So to wrap up, this approach is building on the standardization efforts by the Cami consortium, and is just sort of a plug to say, hey, if you're doing tool development in this area, maybe consider supporting these sorts of input-output formats, because then you might have wider adoption. It can also help you understand some of the benchmarking results that you have. So if you're using Opal and you get a big list of numbers and you ask, so what? It can help you visualize that, do some exploratory data analysis, and better make sense of your data. If you want it, it's up on GitHub. We found out recently that the Conda version was only for Mac OS, so we need to update that to be platform independent, so coming soon. I wanted to also acknowledge Sergey, his postdoc, Jackie, trainee Varuni, in the Cami consortium, and support by the NSF. So thank you.
Question for David? Amazing, Doug. I'll definitely be using the tool. Quick question, and it could be a naive one. Is it so? Does it do taxonomic classification for contigs or for mags, or it doesn't matter?
Yeah, good question. So this particular tool only takes as input a taxonomic profile, and it doesn't care how you generated that. Yeah, so it could be from reads from contigs using k-mers, whatever you want.
And can it only compare two taxonomic profilers? What if I had like 4 or 5 to compare, or that's something it can't take, or something you have in mind?
Yeah, so we thought about that. The pie charts start to get really hard to interpret
Exactly.
with more than
Yeah.
two, so we decided to stick with two.
Okay.
Yep.
Nice. And what happened if they're like 500 or something else? Like the trees you showed were like pretty small. They
Right.
had like 20, 30 nodes. But what happens if it's 10x than that?
Yeah, so a great question. So there's a lot of different command line options
Okay.
to be able to deal with those sorts of situations. So you can specify which particular taxonomic rank you want to plot at. You can filter out ones below a certain abundance. You can give a threshold and say, only show me at most 10 taxa and things like this to try to handle with those situations with really large taxonomic trees.
Amazing. Thanks a lot. Probably gonna get some really stupid questions from me via email, but
Oh, please do. I welcome all questions.
I think there's one from online from Mihai. Does TAMPA handle non-canonical taxonomic ranks? The space between genus and strain is quite more complex than the genus/species strain subdivision implies, and this complexity is highly relevant to some analysis.
Yeah, great question. So support for other sort of taxonomic ranks and taxonomies, yes. So by default, TAMPA will work on NCBI taxonomy. However, it will respect the taxonomy that you give in the taxonomic profiles. So if you want to use a completely different taxonomy, you can. You just turn a certain flag to tell it don't use NCBI, and then it'll respect whatever hierarchy included in your taxonomic profiling input.
I think the last there was a comment from Curtis Hottenhart. As a comment, Graphland isn't integrated with a single profiler. It can take any tabular data and as input.
Yes. Yeah. So to clarify my comment here, the motivation behind my comment saying that's tightly integrated with a single profiler is due to the fact that I could find one conversion script that would convert from that one profiler tool to the input. So yes, definitely, it can take any tabular data. But if you wanted to do it on a different tool besides like a Metaflan tool, then you'd have to do that conversion yourself. That's what I was meaning to say. Yep.